Our first presentation for today comes from TOLULOPE OLAWOLE. TOLULOPE comes to us from the Department of Biochemistry at Covenant University in Nigeria. And here at Michigan, TOLULOPE has worked with Professor Jackie Goodrich from the School of Public Health. And Tolu will talk to us today about her research on the impact of bisphenol A on the health of young adults in Nigeria. Tolu, make sure you switch on your microphone.
Can everybody hear me? Do you hear me clearly? Good morning, everyone, physically present and those that are watching us on Zoom. I'm TOLULOPE OLAWOLE, and I'm excited to be a fall 2021-2022 University of Michigan African Presidential Scholar, and I'm here to make my presentation titled Bisphenol A-Associated Epigenomic Changes in Young Adults in Nigeria. Here I'm hosted and supervised by Dr. Jackie Goodrich of the School of Public Health in the Department of Environmental Health Sciences. I'll be guided by the following outline. I want us to know that our genes, behaviors, and environment play important roles in our health, because the DNA, which is known as the deoxyribonucleic acid, is the molecule that actually encodes the genetic material for every organism. And so the DNA is made up of genes, and these genes go a long way in playing several roles in our health. Genetic changes, for example, are changes that occur to the DNA sequence, but epigenetic changes are, my screen is off. Okay. But epigenetic changes are reversible changes to the gene function, but does not affect the DNA sequence itself. And there are different types of epigenetic changes. One major one that we focus on in our laboratory where I work is the DNA methylation. And so what DNA methylation does is that it adds chemical groups or chemical marks to the DNA. And typically, when the DNA is heavily methylated, it means that the genes, DNA methylation means that the genes are turned off. But when DNA is demethylated, then it means that the genes are turned on. So epigenetics and health, the epigenome, which is the study of changes, of heritable changes to the DNA without affecting the DNA sequence, is susceptible to external exposure during early development. And there are so many things that can cause the epigenome to be susceptible to epigenetic changes. So we have things like, as shown in that image, all of those factors are things that modulate the epigenetic changes that occurs in a microorganism or that occurs in an organism. And the Dutch hunger winter famine, also known as the hunger winter that happened in the Netherlands between 1944 to 1945, has actually proven that epigenetic changes can be passed from one generation to another. So researchers have found that early life exposures have a way of impacting the risk for undesirable developmental programming consequences in generations to come. So for example, it was discovered there that pregnant women that were exposed to the famine had their children having epigenetic modulations throughout their lifetime. Endocrine disrupting chemicals are chemicals or they are compounds, popularly known as EDCs. They are found in a variety of household products. And so they're found in plastics, they are found in medical compounds, even environmental contamination, and they all make their way into our food. So they are found in packaging materials and they find their way into our food. And what these EDCs do is that they have a way of mimicking the actions of hormones. So normally, typically, we will find that we have hormones within our system, but these endocrine disruptors have the ability to bind to the hormones and prevent the hormones from exerting their functions within the system. And so as such, they can end up changing hormone synthesis, the transportation of hormones, and even the binding of hormones to receptors that these hormones should normally bind to. So we are constantly bombarded with these hormonal manipulators. And then it's not just about this, it's not just about at higher doses. Researchers have also found that even at lower doses, these hormonal manipulators go a long way in influencing or contributing to modern health epidemics. For example, people are exposed to endocrine disruptors in children's toys as phthalates. They're also exposed as bisphenol A, which is known as BPA, it's popularly known as BPA, and there are other types like arsenic, lead, parabens. They are found in so many products that are used in household consumer goods. And so low doses even matter. Low doses of EDCs matter a lot, and they contribute to modern health epidemics. For example, like breast cancer, research has shown that low doses or even high doses of EDCs go a long way in contributing to all of these things. And so bisphenol A, as I mentioned, is also an endocrine disrupting compound. So it gets into the body and then it binds to estrogen hormones or estrogen receptors, and it can get into the body majorly through our diet and some other contacts with compounds that are laced with endocrine disruptors. And the lab where I work here, the Do Good Spy Lab, has done a lot of research on the estrogenic activity of BPA, and they've discovered that it has marked effect on the development of rodent models. The way by which bisphenol A acts is that it targets the hypothalamus pituitary gland axis. So that HPG axis usually is the axis where it's like the main center for the regulation of hormone production. So this hormones like luteinizing hormone, follicle stimulating hormones, testosterone, estrogen, and a lot of them are produced via this axis. So since these endocrine disruptors can bind to those receptors, when they bind to those receptors, they prevent the hormones from carrying out their functions, because they will prevent the synthesis of those hormones or the transport of the hormones. And so they can induce cellular responses via this axis. And so there is a growing concern for the presence of these endocrine disruptors, particularly in developing countries. There is really no research going on to target endocrine disruptors within the population. A lot of research has been done in developed countries, and we see that this has influenced policy in so many ways. And for example, you find out that in the United States, there is regulation of bisphenol A in the production of household consumer goods. But in Africa, there is nothing similar to that. And so our team in Nigeria did a review, and we tried to look for, we did a database search for publications that have come out from Africa, focusing on the biomonitoring of endocrine disruptors, and we discovered that only 42 publications in the whole of Africa have focused on the biomonitoring of bisphenol A, not even all the other endocrine disruptors. And that's really, really alarming, because only 20% of them actually focused on biological fluids. And so we said it is actually important for us to be able to understand the environmental exposure profiles in all of these vulnerable populations. And so this led us to this project that we're doing, which is to evaluate the different exposure sources to bisphenol A in young adults in northern Nigeria, and to investigate the relationships between environmental exposures and health outcomes in the selected population. And the aims of this research was to estimate bisphenol A levels in the cohorts that we selected. Then we also wanted to determine the exposure sources. How do this selected population get exposed to bisphenol A? And also, because we know that bisphenol A exerts its action through the HPG axis, it becomes important for us to estimate the association between bisphenol A concentrations and hormonal profile. And also, we wanted to, because we know that bisphenol A causes epigenetic changes, it was important for us to estimate the association between bisphenol A concentrations and DNA methylation at some genes. And so we recruited LD Covenant 120 Covenant University students. So we did that because it's more of like a controlled population. The students stay inside the university and they all live there once they come home, or once they come to the university. And then we administered questionnaire and we collected whole blood in vacutainers. We got plasma and DNA from all of those samples, and we also collected urine in BPA-free bottles. And DNA was isolated from these blood samples, and BPA was estimated in the urine that was collected, and also the hormonal profile was done in the plasma. And here at the Do Good Spy Lab, I worked on bisulfite conversion and pyrosequencing, which is focused on DNA methylation at some environmentally responsive genes, which are those 3 genes that I mentioned here. And so what pyrosequencing does, because that is like the main thing that I did here in this laboratory, is that it's like a standard, is considered a gold standard for assessing BPA, for assessing DNA methylation in DNA methylation status. And so what it does is just that it freezes the methylation status at some very rich CpG islands. Some of us might not really be familiar with that, but it's considered a gold standard for measuring BPA, for measuring DNA methylation. And so the statistical analysis was also done, and the exposure was like, the exposures were bisphenol A and the dietary sources, and the outcomes were the hormone, the DNA methylation, and also BPA was also considered as an outcome when we're looking at the dietary sources. So there was a lot of results, but for the sake of time, I'm only going to present a very few and interesting of the results that we found. So this is just the descriptive statistics or characteristics of the study subjects. Most of the population were males, and we also discovered that, of course, BPA was detected in all the urine samples that were collected. And then, as expected, we found out that testosterone and testosterone levels were higher in males compared to females. And we also found out that hormones like prolactin and follicle-stimulating hormones were higher in females compared to males. And so in estimating the BPA levels, it was interesting to find out that in this particular cohort, the BPA levels were actually lower compared to what was obtainable across other continents. So even though this sounds good, but we have come to also realize that low doses of endocrine disruptors still go on to perform the same, still go on to like do things that you wouldn't want them to do. So it's not the fact that it is detectable in the population is also a signal to the point that we need to do more to be able to consider other exposure sources. And then we also tried to look at the impact of eating habits on bisphenol A, and we discovered that females that ate breakfast, lunch, dinner, and also ate in plastics had more BPA concentrations compared to those who didn't. And we also saw that the eating pattern behaviors of males versus females also had an impact on the exposure sources to bisphenol A. And we all tried to find the association between bisphenol A levels and the hormones, and it was interesting to see a lot of things there, even though in the male. So here we tried to do a sex-specific response model, and we saw that even the males, for example, bisphenol A did not have any correlation with all the hormones, but we found out that some of the hormones were correlating with one another. And this is a pointer to the fact that we know that bisphenol A acts through the HPG axis. So even though we might not find an association between bisphenol A and all of those hormones individually, the fact that these hormones are interacting with one another is a pointer that we need to take note of. But even in the females, we still found some association. And so environmental exposure to bisphenol A has been reported to actually cause increased serum levels of some of these hormones. And that is similar to some of the things that we found in our research. And then the genes of interest that we looked at were these 3 genes. And what these genes, the reason why we looked at these genes is because the production of some of these hormones like progesterone and estrogen actually go on to activate the DNA, to activate the transcription of these genes. And these genes play a lot of roles in estrogen-inducible, they play a lot of role in growth, metabolism, and sexual development. And so what we found here was also very, very interesting, because at this point now we saw that bisphenol A was correlated. There was an association between the levels of bisphenol A and the methylation of some of these genes. And so research has shown that higher bisphenol A concentrations can be linked to hypomethylation, meaning that it reduces the methylation status in females. And so this was similar to what we actually found out. And so, in summary, we tried to estimate BPA concentrations in the cohorts, and we were able to detect BPA, to detect bisphenol A in the samples that we assayed for. And even though the BPA levels were lower than than what was obtainable in other continents, it's still a shocker, or it's still something that should be looked into, because we know that low doses of bisphenol A also matters. And in determination of the total exposure sources, we discovered that the major exposure route to bisphenol A in this studied population is via diet. The eating habits of the the eating habits of within the groups impacted BPA levels. And we saw that eating in plastic is a primary predictor or primary source of exposure to bisphenol A. And we were also able to estimate, in estimating the associations between bisphenol A levels and the hormones and the DNA methylation, we saw a relationship and a correlation between all of these genes. Even though we didn't see significant associations within the models, but we are planning to do to investigate sex-specific models. That means that we're going to like stratify the results by sex and then see what we're going to find out. And so in conclusion, we had some limitations in this study because we were limited in resources, we were limited in funding. Our sample size was small because we could only get like 120 samples because this kind of research requires a lot of funding. And so we are thinking in future directions that we're going to increase the sample size so that we can validate this in a larger population. And so what should be the take home for some of us that we are listening to this talk? It's about the public health implication of this research. And awareness needs to be created in developing countries so that individuals can make better choices to reduce the exposure to bisphenol A. They have to know that microwaving food in polycarbonate plastic food containers, it's not healthy. And so there are containers, these containers, all our containers always have numbers under them. So it's good to avoid plastic containers with the number 7 because those ones are made of polycarbonate, and that's bad, and that's one source of bisphenol A. And also, we have to reduce the use of canned food because the lining of the coating of the cans are made of bisphenol A, except it is explicitly stated on the cans that they are BPA free. Some of our water bottles, some of our canned foods have it on them that they are BPA free. And also, as much as possible, it's just better to eat fresh foods. And when possible, use glass, use porcelain, use stainless steel containers, particularly for hot liquids or hot food. And most importantly, for children, it is good to avoid the use of formula bottles or toys that contain BPA because research has shown that over the years, there is that it increases the susceptibility to developing diseases in the nearest future. These are just more public health implications. Never reheat your food in plastics and avoid the use of food wrapped in plastic markers. I would like to acknowledge the University of Michigan African Studies Center for the privilege given to me to come here and learn, to be able to work in the laboratory where I worked. I would also like to acknowledge the university where I work in Nigeria, Covenant University, for giving me the platform to be able to be here. I would like to acknowledge my host, Dr. Jackie Goodrich. She's been wonderful. She's really made my stay here very comfortable, and she's been guiding me through the whole process. I would like to also thank Professor Dina Dolinoy, who is the head of the laboratory where I work, the Do Good Spy Lab, and also the chair of the department. She is more than awesome. And Rebecca and Jessa were the ones I shadowed to be able to learn the techniques in the pyrosequencing. My acknowledgement goes to them. And then the best lab in the University of Michigan, which is the Do Good Spy Lab, the lab where I worked. That's a picture of us at our last Christmas party. The lab has been wonderful. Everybody made me feel welcome. And I would like to thank my UMAPS 2021 cohort. You guys made the period so relaxing. It was just like family. It was just like being in the midst of family. And my special thanks goes to the Nigerian team that I work with, Associate Professor Solomon Rotimi, a senior Fulbrighter, and Dr. Kemi Rotimi. She's also a UMAPS alumni, and Dr. Pedro Campos and Dr. Isaacson Adelani. I would also like to appreciate and acknowledge my husband, who has been a very strong supportive system to me. He's the one that's been keeping the home front, taking care of my daughters, not making me to feel my absence from the home. I'm really, really grateful for all of this. Thank you so much for listening.
Thank you, Tolu. That was perfectly timed and very accessible. Thank you. I mean, I'm not a scientist, but I actually think I understood the main message. So perfectly done. Thank you. So we have time for questions. So anybody from the audience, I have microphones here, since we're also broadcasting on Zoom, and we have about 30 people at the moment. If you ask a question, please use the microphone so that they can hear you. Lare. And We only have 10 minutes, so please keep your question short.
All right, thank you very much, Dr. OLAWOLE, for that presentation. Very accessible, and even though I'm not a scientist, but I could understand a lot of the facts you shared with us. One question that I have is, considering the limitations of your study, particularly the fact that it is limited to the campus of Covenant University, and also the fact that I'm assuming many of the students in this university are from middle-class families. So if the same study were to be done outside of the campus, particularly in the community that is very close, in close proximity to the campus, will the outcome be different from what you found? And the reason why I'm asking is some of the conclusions you came to, particularly the idea that canned food, either in plastic and other things you mentioned, are not things that are easily accessible to those who live outside of this community. Many of these communities will maybe eat fresh food, probably from their farms. So will the result be completely different if you were to use those communities as a sample for your study?
Okay, thank you very much, Prof, for that question. That was a very good question. So I still believe that the results will be very different. Well, I can't tell until we're able to like do something. But the essence of it is that we're starting with this controlled population before then we'll consider moving out. But one thing I must say is that bisphenol A has been detected in water sources in Nigeria, in Africa. So even though the community out there might not be taking the luxury things like canned food, but we still have other sources of bisphenol A that is not just solely based on canned foods. For example, the plastics, and the reason why this awareness needs to be created is that the plastics that are made, there is no regulation for the usage of these compounds in plastics in Africa. So you come here, I think Bumi's bottle has BPA free. You don't find that in Africa. Before you can get containers that are BPA free, you really have to pay a lot of money to get them shipped in. So, but the community outside, they're still exposed to other sources. So I still believe that the results, there will definitely still be something interesting. But of course, funding has to be available to be able to do that kind of research. Thank you, Prof. I hope that answered your question.
Yeah, sure. Anybody else? Enrique. Yeah, why don't we, also for the sake of people on Zoom, say your name when you ask a question. Our first question came from Professor Omalade Adunbi. Now, Enrique. Speak up a bit.
Kind of answered it, because I was interested in policy, and you told us, in terms of personal choices, what we needed to do, and you just mentioned there aren't any requirements that there's BPA-free products, and I know it's different between the U.S. and Europe. Do you know of any countries where this is on the table as a policy recommendations to ban the use of bisphenol A?
You mean countries specifically in Africa?
Oh, right,
or generally?
in Africa, yes.
In Africa?
Yeah.
No, there is no country in Africa that has that under any form of regulation, because we did a review, and so you can also actually look up that review paper, is on the internet. We did a review and we discovered that there is no form of regulation, because you can only regulate what has been researched upon. So if there's no research to back up that regulation, then there is nothing to regulate. But there's a lot of regulation in developed countries. I know in the US, bisphenol A has been banned in the production of baby bottles and a lot of other containers too.
I guess I was asking not if the regulations are on file, but if there's interest in making policy.
I wouldn't know if there is any interest until we get the research out there first.
Good.
Thank you.
Do we have any questions on Zoom, Enrique?
No.
Well, then I will use my prerogative as the moderator and ask you a question, TOLULOPE. So you mentioned that you found, if I understood, lower levels in your study than the studies found in Durban, South Africa, and elsewhere in Africa. Do you have any idea why that is?
Okay, thank you for that question. So the studies that were done in other parts of Africa there was one that was done on mothers and child care. And so that's another project that I'm working with Dr. Jackie Goodrich on. So, but this is the first time that we're looking at that BPA concentrations in like young adults. Mm-hmm. So all the studies that have been done has been on children or pre-adolescents. And so we might not see, like the findings might be very different, but we cannot conclusively talk about that now until we're able to validate it in larger sample size.
So we do have time for one more short question, if somebody has a short question. Katie Wandoff. So introduce yourself also.
Hi, I'm Katie Wandoff. I'm just curious what you are going to be doing when you return back home, and how you're going to use this experience to further what you're doing there.
Thank you very much. That's a very big question, because there's a part of this research that was done in my university in Nigeria, but the part about the DNA methylation was done in the Dugot Spy Lab. And so fortunately, I have a working relationship with the laboratory, and we're looking at applying for some funding to be able to expand the sample size so that we can make conclusive information. We can get conclusive information about the research. But when I go back home, definitely we're going to try to recruit more people into the study, because we need to first get this research out there. When we get this research out there, then people can know that before this becomes a very big blown problem. Like now in the United States, there are issues of environmental pollutants like the PFAS, the phthalates, all of them. People are looking into them. So we don't want it to get to that point in Africa, and so that's what I'm going to be focusing on when I get back to Nigeria.
Thank you, TOLULOPE. Very nicely done. And that was kind of a perfect question on when to end, on what to end in this presentation, right? Because that's one of the things that we've seen happen so often with the UMAPS program, how the collaborations continue after the end of the program. So we're very happy to hear that's already in the planning for this particular project.